All right, our next speaker is Molly Smith. Molly Smith has three faculty members, John Beck, Alan Garvey, and Brian Degenhart, and she's going to be talking about manipulative treatment data analysis.
As you said, I'm Molly Smith. I'm going to be a senior CS major here next year. And our project is analyzing, mining, and modeling medical manipulative therapeutic data for quantification and reproducibility. So before we begin, I'd like to give just an overview about what I'm going to talk about today. First of all, I'm going to give you a background on information, a little bit about AT Still Research Institute, a little bit about manipulative medicine and their lab setup over there. And then I'm going to talk about the goals and objectives I have for this semester, for this summer, and the methodology that I'm going to be more developing during the summer, and the significance of the project. To begin, my project is basically stemming out of ongoing research done at AT Still Research Institute, and more specifically the Center for Advancement of Osteopathic Research Methodologies, or CORM. Last April, they received a $4 million endowment to do this research, and what they're investigating is, first of all, they're investigating the reliability of osteopathic diagnostic tests. It's basically they want to know that every time that you do a test, you get the same results. And in addition to that, they want to know that every physician that does this test gets the same results. And in addition to that, they want to know the validity and accuracy of the results. So now we're getting the same results every time, but we want to make sure that those are the right results. And they also want to know the clinical relevance of this, how relevant is manipulative therapy to modern medicine. And so basically the whole overall project is just going to provide an evidence base that it is so important to the future of osteopathic practice. And how they plan on doing how it's going to be done is to develop standardized methodologies and new tools to quantify manipulation. And so to give you an overview of manipulative medicine and some of the words I'm going to be using, basically, manipulative medicine is just a tool that practitioners use to first identify different structural characteristics and then treat those structural characteristics. And basically, it's a way for osteopaths to provide that patient-specific care that we're looking for. There's two types of manipulative medicine, so two sections. First of all, there's the diagnosis, and then there's the treatment. I'm going to concentrate mostly on the diagnosis. So first of all, what they're looking for in the diagnosis is some somatic dysfunctions. And there's 4 types of somatic dysfunctions, and they're represented by the acronym HEART. So the first one is tissue. And so basically they're looking at the tissue, any abnormalities in the tissue. They look at skin to deep muscle tissue. They talk about balling of the tissue and things like that. The next one is asymmetry. Basically, they want your body to be symmetric. They don't want any kinds of asymmetry in the structure. And then they also talk about restriction. So any kind of restriction in movement is important to know. And then they also talk about tenderness. This is actually important to note that this really isn't pain. It's reaction to palpation. And so it's really important to say that the palpation is important to finding tenderness. And in this current research, they're basically concentrating on the diagnosis, and within those somatic dysfunctions, they want to quantify asymmetry and restriction. And to do this, there's been developed a 3-stage system. It's basically a crawl, walk, run system. The first stage, the crawl stage, is where they use these paper models. Basically, it's just a piece of paper that has lines on both sides of the paper, and the practitioner or student will put their thumbs on these lines, and they'll say, my right thumb is 3 millimeters superior to my left thumb, or something like that. And then they'll be able to quantify how close they are to the right results and how many times they'll get those right results. And then the next stage is the use of anatomical models that they've used. And so basically this is also assessing asymmetry. They'll put their thumbs on certain spots and be able to tell how symmetric it is. And actually with these models, when they're actually being used, they're covered up so they're more realistic. And then the run stage is actually the use of human participants. So basically this is going to be used for education and future DOs to give more quantifiable training, and also through osteopathic research. And it's going to provide that evidence base for manipulation that's so important. So the lab setup over at ATSU has a few different kinds of equipment that collect data. The first type of equipment is this 2D camera that shoots straight down at the OMT bed. And the next piece of equipment are these 3D cameras that they have that that track the thumbs of the practitioner. And also they have these pressure sensors that track how much pressure is being used during palpation. So basically the 3D cameras are used to first line up the patient correctly. So during treatments, they want to make sure the patient is first lined up correctly so that they can check to see whether the rest of their body is symmetric or asymmetric. And it's also used to test asymmetry. The 3D cameras are basically used to test asymmetry also based on the positions on the practitioner's thumbs. And pressure sensors are used to assess restriction. So basically, the data that these equipment collect is pretty ugly. This is just a really small set of what it does. It collects three locations of both of the practitioner's thumbs, which is based on the refraction points on the thumbs. And as you can see, those are the points, those gray points right there. And those are in different parts of the hand, but mainly we're concentrating on the those are the thumbs. And the pressure sensitivity looks a little bit like this. This is just one snapshot of a pressure sensor at one point in time. It's a 64 by 64 matric of basically the pressure sensors have little tiny pressure sensors in them that collect the data. During one session, we can get between 200 and 250 matrices. So now I'm going to go over a little bit about the goals and objectives I have for the semester. My first goal is to automate the model calibration. So basically, these models move in two different directions. The first direction they move is the shear direction. So basically, there's two plates. You can see this is one plate, this is another plate. And basically, the right plate will move either 1, 2, or 3 millimeters forward or 1, 2, or 3 millimeters backwards. And they want to be able to make sure that they know exactly what places the specific points of interest are when those movements are made. And then also, it rotates 1, 2, or 3, or 1, 2, or 3 like that. So the refraction points, they put the refraction points on this model on the ASIS, the PSIS, the cubical, and the iliac crest. And just to give you a better idea what those are, just ASIS and the iliac crest is this whole thing, the PSIS and the two cubicles are right there. And one thing that they can have trouble with in the model calibration is these two spots right here are so close together that cameras get confused. And so I'm going to be spending time trying to figure out how to make sure that that doesn't happen. And then basically, just the whole automation will be important to them just because calibration, just the analysis part takes 6 to 7 hours for a person to do it. And that's not even including taking the pictures of the models. Next goal I have is to identify palpations in the 3D camera data. This is just a graph of the 3D distance moved on just one of the thumbs. So basically this is a graph, and it's a pretty ugly graph, but basically the human has to go in and decide where palpation started and where palpation ended. And so we want to be able to, and these are the points that they selected. So you can see like that's what a palpation would look like. And this is a zoomed-in picture of it. Basically, we want to be able to figure out a way to find those, automate that process, and be able to find those. The next goal I have is to also identify the palpations in the pressure sensor data. And so this is a picture of what the pure pressure data looks like. And so if you can see, there's all this various data that needs to be illuminated. And also, the area of the thumb needs to be found where we actually think that the pressure is And the next step we need to take is to merge these two data streams. We have the 3 camera sensor data and the pressure sensor data, and we need to combine those to get the same results. Some things that we'll have to look at is the left hand versus the right hand data. So basically, we need to make sure that we're finding the same results when we find the same populations in both left and right hand. And then also we'll have to deal with the fact that data is collected at completely different intervals. The 3D camera sensor collects data at 30 times per minute, and the pressure sensor collects it at 162 times per minute. And our final goal, and this might be a future goal, is to develop some kind of visualization of this feedback and give not only a visualization, but a quantifiable visualization. So basically, we need to tell the students or practitioners how close they are to the correct answer and how reliable they are. And we also want to make sure that they're displaying information from both sources. Currently, the only kind of visualization they have of this data is just a picture of the pressure. And it'd be really important to give that feedback to the students and practitioners in a different way. Now I'm going to go through a little bit of the methodology that I'll be using this summer. Basically, I'm going to be using MATLAB extensively for the analysis and trying to find the patterns of populations in the data. We've been using it mainly because it has many built-in functions that I feel like will be useful, and it contains extensive graphics features that will help me visualize these large data sets, and as well maybe use it in visualization later down the line. I'll also be using Perl to, first of all, for the calibration of the models, and maybe down the line after MATLAB has helped me understand what the data is saying. To give a little bit of significance on the project, basically this whole project is just providing this evidence base that everybody's wanting. When you go to the doctor, you want to know that this works. You want proof that what the treatment that you're getting works. And so basically, currently physicians are more and more leaning towards using treatments that have been studied and have quantifiable results. And basically, currently there are no objective tools to measure manipulation. Basically, it's all apprentice-based. An expert does the manipulation. A student will then do it themselves, and the expert can say, okay, you did it right. You did it wrong, but you don't really know how wrong you did it. And then you need quantifications for the skills of experts and trainees. And then for more of the significance of my part of the project, immediate feedback is necessary for the educational and training aspects of the project. Currently, a student will do a publication exercise, and then they'll get results a week or 2 weeks later, and they forgot everything that they've done. So it's really not useful at all for that kind of feedback. So that's the big significance in my part of the project. The overall goal of the bigger project is to develop and pilot these instrumentations and develop new standardized methodologies for manipulative research, and just give validity to these osteopathic palpatory tests. And mine is just basically to analyze mine and model the data to give immediate feedback from that quantifiable feedback. So, and I want to thank my advisor, Dr. Garvey, and Dr. Dagenhart from ETSU, and also Chin Young, Jane Johnson, and Brennan from ETSU.
We do have time for questions.
I'm really great as well. Can you give me either a hand example or is it general description of one of your palpitations?
Palpation, what they're describing as like one palpation is basically a 1, 2, like 1, 2, 1, 2, 1, 2, 3 times. And so that's with thumbs. And that was, that's basically the most basic palpation that you can do. But the data that I showed you, that's what it was, is the 1, 2, 1, 2, 1, 2. And it's always 3 times in a row.
I was wondering how you learned what you learned about manipulative medicine so far.
From these people at ATs. I actually just learned a lot about what my project's about and the background information on the whole project. This week I went and met with Janine and Jane and Dr. Dagenhart was on the phone, and so I learned a lot of that. Basically got a lesson in osteopathic medicine.
Yeah. So were you observing them working on patients, or?
Well, the first time I've been there, he actually Dr. Dagenhart actually demonstrated the equipment. He demonstrated palpations and things like that. And then this last time they showed me the models and how the models worked and what the data actually showed. For a long time, I didn't even understand the data because I didn't understand the manipulations. And now that I can do that, I'm going to be able to develop new methods to actually work with the data. So.
Well, I'm just curious about something. You mentioned in one of your early slides that there was a $4 million, maybe you should turn it down.
Yeah.
Who's that from?
$2 million is from the Osteopathic Heritage Foundation, and it's in honor of 2 graduates from the osteopathic school here in town. I'm not positive if I can figure it out, but and then that was matched by ATs.
So I'm curious, when using this to help a single trainee learn, are they trying to use it to help eliminate bias between both hands, or to get a consistent amount of pressure, or what exactly are we looking for with this pressure of this?
It's looking for reliability and accuracy. So basically, you want to make sure that you, like we said, between physician to physician, you want to get the same results. And so being able to quantify how right you are, like say that you think that your thumb is 3 millimeters above your other thumb. And this is a symmetry, that's the somatic dysfunction asymmetry. So you think that your right thumb is 3 millimeters above your left thumb. Well, maybe that's not even true at all. And so if you can have quantifiable feedback saying you're only 2 millimeters above your left thumb, then that gives you a stepping stone for being able to get it right the next time. And give that consistency between physicians, between treatments, and so it's basically the pressure. I'm going to learn more about the pressure data later once I get the system working right, but that's that general idea. So that's
do you know if there's any plan then to take these data that you're collecting and to then correlate them with the outcomes of the treatment? To say, well, if the therapist is actually off by a certain amount, does that actually make any difference in terms of outcomes? So are we clear that, that this treatment really does need to be this precise, or is it possible that, that it doesn't, that there's not quite as much precision needed?
I am not positive on the answer, but I know that they are looking at how relevant they are. Like even if the outcome of this specific test doesn't really change the structure of the body, then it's not relevant in medicine, in modern medicine. So they are looking at how it changes. I'm not sure if they're looking at the actual precision of how precise you have to be to make that change. But I know that they are looking at whether it's relevant. Does it work? So maybe you 2 millimeters and 3 millimeters doesn't matter, but I know that is the measurements that are taken and how specific they are being. So.
You mentioned that you've been doing some data mining.
Mm-hmm.
What are you going to be looking for?
In the 3D data, I'm not really sure what I'm looking for yet. That's the first stage that I need to take, but I'm going to be looking for where the palpations start and where the palpations end. And so that way that they can, basically, the first stage that they want us to get to is to be able to give a report of statistics based on the sessions. And so if I include all of the experience data and all of the stuff in between palpations doesn't really tell me anything. So basically, I'm going to be able to need to find those specific points. I don't know how or what those specific points are going to look like, or yeah, that's something.
Maybe. Back to the So are you going to like have your model physician that you base this on? Here's what I'm trying to say. If you're looking at these compounds, these palpitations, and you're trying to look at how little variation there is between people, who's going to be the perfect, who has a perfect compound that you're trying to, does that make sense?
Yeah, I understand what you mean. Like, what's right? What's the accurate answer?
How do you know, who would you use as your
Well, basically, for the accuracy, they can't really do that on the human yet, so you can't really tell how accurate you're going to be. But that's why they're using those models that I showed. These models. So that can show accuracy. So basically, they're calibrating these with 3D data, and so between physicians, they can tell how much reliability there is based on this accuracy. So they can't really do that on people yet. I know that's they were talking about the fact that currently with a human being, all they can do is consensus testing. All they can do is say, okay, we have these 10, or however many experts they have in the field, and they all come to the same answer. So that's what they consider accurate on a human being. Whether it is accurate or whether that's up in the air still.
So that means you're actually more interested in consistency. So you want something that is less variable. Does that make sense? Less variance.
I think the variability is real important, but I think they're still looking for that accuracy. And like I said, it's basically with the model training that they're looking at that. I don't think that variance is less variance is more important than accuracy, but that'd probably be a question for what Dr. Degenhardt. But that's my understanding.
So whenever we come up with a mathematical or computational way to describe something, there's always a second step: validation, process validation, or model validation. When you have your method for understanding where palpitation begins and ends and whatnot, have you thought about how you're going to validate your method?
Basic validation is going to be so Chinning is the person at HSU that basically does all of these, finds these palpitations. And so currently the only validation that I've thought of is that she, what she picks out is what I want to find. And so if I can find a method that finds these and then she goes back and she picks the same ones, that's the only real way of doing it, because there's really no other measurement of what a palpation looks like, other than what a human says it is and what a human knows it is. So
you might also consider getting an expert to do a model palpitation, so you know exactly when one's supposed to begin and how much pressure there was at various places. So you know going into it what the data ought to be looking at. And that's another way of using, instead of using experimental data, but dummy data.